Thank you so much. And first and foremost, I'd like to start by thanking the organizers for putting together this fantastic symposium, highlighting so much beautiful science. And today I'd like to share with you the story that I've been working on over the past couple of years. I'm a postdoc at the Vamsi Mutha's lab, and it's a joint venture both at the Massachusetts General Hospital and the Broad Institute. And I'll start with a confession. I am fascinated by the mitochondria. And so here you can appreciate these beautiful intracellular organelles dancing in real time within a living eukaryotic cell. And I'm enamored of them, not just because of their beautiful dynamics, but also because they're hubs of key cellular functions within the eukaryotic cell. And so they control things both linked to energy production, but also metabolite production, cofactor synthesis, as well as many cell fate and death and life decisions. And so my work focuses on the most common monogenic mitochondrial disease that affects 1 in 50,000 people. So there's thousands and thousands of patients of this disease worldwide and even in Israel, and it's called Friedreich's ataxia. So what is this monogenic disease linked to? One specific gene, a loss of expression of the protein ultimately frataxin, which ultimately encodes for mitochondrial protein. So what happens in this disease? Usually in healthy people, the frataxin gene within its first intron has a GAA repeat that can be somewhere between 5 to 20 repeats in length. But in these individuals, you have a massive expansion of the GAA tract so that the GAA tract is now somewhere between 60 to tens of hundreds of repeats long. And through several different mechanisms, ultimately what this cascades into is that there's reduced frataxin mRNA and protein levels so that there's at any given tissue in these Friedreich's ataxia patients, very, very low levels of the protein, somewhere between 5 to 30% residual levels, even though I should note that this protein in Friedreich's ataxia is completely unmutated. So it's a completely functional protein just expressed at very low levels. Now, how does this disease present itself? The first thing that these patients typically come to the clinic with is ataxia, and that means that they've lost coordination of their hands and legs in space. And this is a progressive ataxia, and as they get older and older, it usually presents in late childhood, early teenage years. It'll get progressively worse so that by 10 years post-diagnosis, they're oftentimes wheelchair bound. A second feature of this disease is that it's oftentimes linked to diabetes. And so there's beta cell dysfunction. And then ultimately, these patients succumb to premature death at around 35 years of age due to cardiomyopathy complications. And the thing that I'd like to highlight here is that currently we have no proven therapies for Friedreich's ataxia. So there's a lot of supportive care that can be offered for these patients, but nothing really therapeutic. And so what does this protein frataxin actually do in the cell? As I said before, it functions in the mitochondrial matrix. And here I've highlighted it for you here in blue. And frataxin works together with several other proteins, forming a complex that ultimately will take sulfur and iron and an electron and generate these essential ubiquitous cofactors called iron-sulfur clusters. Now, iron-sulfur clusters in human cells come in 2 flavors, either 2 iron, 2 sulfur, or 4 iron, 4 sulfur. And they're incorporated into a wide variety of proteins, and they can do many different things. But the most prevalent one is that they're electron acceptors. So sometimes they're called the wires of the cell. They can really easily accept and donate electrons. And they're incorporated, as I said before, to a wide range of different proteins, and they're essential for their functionality. And so we can find them in machineries that deal with DNA replication and DNA repair. Also, things like nucleotide biosynthesis, ribosome release, and tRNA biosynthesis are known to be dependent on iron-sulfur cluster containing proteins. And they're also take place in more mitocentric functions like respiration or the TCA cycle. So as you might begin to appreciate, in the absence of frataxin, which is an allosteric activator for generating iron-sulfur clusters, all of these processes in the cell come to a grinding halt. And so the premise for my work was trying to find a therapeutic for Friedreich's ataxia. And we initially were inspired by an evolutionary link that we found in the literature, which is that iron-sulfur clusters evolved in very, very oxygen-poor environments. And this, together with our lab's historic interest in the link between oxygen and mitochondria, led us to the question, how would oxygen affect the growth of frataxin cells? In other words, if we take frataxin cells and now place them back into this evolutionary cradle of low oxygen conditions, what would their phenotype be like? And so we took human cell lines. In this case, you're looking at K562 cells, and using CRISPR-Cas9, edited frataxin using one of two guides. You can see that we deplete frataxin by Western blot pretty effectively. And then what the first and most obvious thing that happens in these cells is they take a massive hit to their growth rate. And this has been described many times before in the literature, and it's always been done at 21% oxygen, which is the ambient oxygen tension that we currently always breathe. Now, what happened when we did this same exact experiment but placed these cells under 1% oxygen? We were astounded to see that even though these cells were significantly depleted for frataxin, they seemed to be completely impervious to the fact that they no longer had this protein on board, and they were growing just as well as their wild type controls. This sort of turned the dogma on its head for us for whether frataxin is essential for viability or not. But we wanted to take this one step further and ask this in a more multicellular organism where multiple different cell types have to function correctly in order for the organism to develop and bear fruit. And so in a very fruitful collaboration with a fantastic postdoc, Josh Meisel at Professor Gary Ruvkin's lab, we tested this exact same question in C. elegans, the nematode. So Josh took these C. elegans that are deleted, fully deleted for frataxin, and then cultured them under 21% oxygen. So what you're looking at here on wild type is worms crawling around the plate happily, devouring the bacteria and moving around. But these frataxin nulls were very, very stunted for growth and halted at a very early stage of development. But if Josh did the same exact experiment, simply place these frataxin nulls under 1% oxygen, what he saw now was that these worms could fully complete their development. And moreover than that, they could even generate progeny so that they can fully recapitulate their entire life cycle, even in the absence, the complete absence of frataxin. And so what we saw was that frataxin human cells, yeast, I didn't have a chance to see this, but to show you this, but we also saw this in yeast and even worms. These full nulls are completely viable in hypoxia. And it sort of flew in the face of conventional wisdom that frataxin is an essential protein. So what is the mechanism of this rescue? A priori, we could come up with several hypotheses. The first one is that we know that there's altered transcriptional states in hypoxia, and perhaps something about this altered transcriptional rewiring somehow makes frataxin unessential. The second thing that we thought was maybe iron-sulfur clusters are simply indispensable under hypoxia. You don't need the machinery because you don't need the clusters themselves. And the third hypothesis that we could come up with was that hypoxia directly boosts cluster biosynthesis in the same way that frataxin, the allosteric activator, boosts cluster biosynthesis. Now, to cut a very long story short, I'm going to tell you that the first two hypotheses did not bear out. So these first two things don't seem to be playing a role in how hypoxia is rescuing for the loss of frataxin. But the third one, when we looked a little bit into it, was very, very intriguing. So to look into this, we partnered up with Sachin Patra and David Barandu, who do these beautiful in vitro reconstitution reactions with just isolated components of iron-sulfur cluster synthesis. And we asked them to run this reaction with the human proteins and do this either with and without frataxin or with high oxygen tensions or low oxygen tensions. Now, what did they see when they ran this reaction? First, what you're looking at here is cluster synthesis rates. If they ran this reaction at 21% oxygen with and without frataxin, we saw a dramatic shift with frataxin. This is totally in line with the fact that it's thought to be an allosteric activator of iron-sulfur cluster synthesis. But surprisingly, when they ran this reaction now at very, very low oxygen tensions, what they saw was that even in the absence of frataxin, you had significant generation of iron-sulfur clusters. And this seemed to be sort of a compound effect that both frataxin and hypoxia are independently boosting up cluster synthesis in parallel ways, because when they had both of these components on board, you had an even higher rate of cluster synthesis. And so just to summarize what I've shown you here so far, frataxin is a known allosteric activator of iron-sulfur cluster synthesis, which is important for mediating many essential processes in the eukaryotic cell. It seems like hypoxia can completely bypass further loss of frataxin directly by boosting the exact proximal step that frataxin affects, namely cluster synthesis. Moreover, through additional experiments that I don't have time to show you today, we could show that hypoxia has another mechanism of action of rescue, and that it seems to also be boosting the levels of the substrate, bioavailable iron. And so with this mechanistic understanding at hand, we wanted to turn to sort of the ultimate preclinical question, and that is, would hypoxia affect or have any benefit for Friedreich's ataxia mouse model? And we were very fortunate that at the time that we were thinking about these questions, a fantastic mouse model had just come online that recapitulated many of the key features seen in Friedreich's ataxia. This is a whole body knockdown where you give the mice doxycycline and it induces an shRNA against frataxin in every single tissue of the mouse's body. So what we decided to do is take this Friedreich's ataxia mouse model and house it under one of two oxygen tensions, either normoxia, which is 21% oxygen, or hypoxia, which is 11% oxygen. And with 11% oxygen, people always take a step back and are a little scared when I say this. I should note that 11% oxygen isn't the top of Everest, but rather it's base camp, and it's a tension of oxygen that people can live with and supports human physiology for long term. And then we tested the proximal neurological deficits, which are the key features of Friedreich's ataxia. And we did this by one of several methodologies. Today, I'm only going to be showing you two: inverted screen, where we test the ability of the mouse to grip, and rotorod, where the mice are walking on a faster and faster rotating rod. And both of these things test features that are oftentimes lost very early in Friedreich's ataxia. So what did we see? We saw that, first of all, the mice that were housed in 21% oxygen had a significant development of ataxia compared to their wild-type controls at 21% oxygen. But the simple intervention of housing them at 11% oxygen now significantly blunted the deterioration of ataxia and had a notable effect both at early and late time points of the disease manifestation. So just to summarize everything, it seems like hypoxia has a clear benefit to the absence of frataxin and can alleviate and boost the synthesis of iron-sulfur clusters. And this in turn has great therapeutic potential for multiple modes of, or multiple models of Friedreich's ataxia, be it cell culture, yeast, nematodes, and even mouse. And with that therapeutic potential at hand, we're currently looking into more practical and safe regimens for patients. With that, I'd like to end and thank the people who were so instrumental for this work. So first and foremost, my mentor, Vamsi Mutha, who is such a fantastic leader in this, pushing this forward, this project forward. My collaborator, Josh Meisel, who did a lot of the C. elegans work that I didn't have time to show you today. The people in the lab, as well as our collaborators and my funding sources. I'm happy to take your questions.
Thank you, Tslil. We have time for one question. So Professor Gross.
Hi, Tslil.
Hi.
How are you?
Good. How are you? It's good to see you.
I'm great. I'm just, first of all, I want to apologize that I didn't hear your talk when you came to Weizmann or virtually came to Weizmann. And second, I send my love to Vamsi.
I will.
And the question is, actually, I was thinking, why nothing about glycolysis and oxphos? You know, I would think that maybe under hypoxic conditions, these animals would use glycolysis instead of oxidative phosphorylation because they don't have the clusters of at least the respiratory complexes. But you didn't say anything about that.
That's right. I sort of blazed through everything and gave you just the punchlines, right? Okay, so I'll just say first of all, it seems like hypoxia, when you put these Friedreich's ataxia models in hypoxia, they're boosting up the levels of the respiratory chain that you're right is deficient in 21% oxygen. So they're completely able to restore respiration, and that balance between glycolysis and oxphos is most likely restored. But moreover than that, I'll say that the deficits that we see, for example, in growth, seem less to be linked to the problems with respiration and more to do with things that have flavors of DNA regulation. So for example, we know that in the absence of iron-sulfur cluster synthesis, you have a blunting of many of the DNA polymerases as well as the DNA damage response. And both of these things are restored under hypoxia.
So there's no metabolic rewiring in these patients or in these mice?
There's clear metabolic rewiring, but because hypoxia seems to be directly affecting the step of cluster synthesis,
Yes.
you're having a restoration of all of the downstream cascades.
Amazing.
Okay.